Good morning everyone, welcome back. So last class we have discussed about Mendel's law of inheritance, right? We have seen Mendel's law of dominance, Mendel's law of purity of gametes and Mendel's law of independent assortment. So we have seen monohybrid and dihybrid ratio, phenotype as well as genotype, right? And we have discussed exception of Mendel's law of dominance, right? That is incomplete dominance, right? So in complete dominance, we take the example of 4 o'clock time. Okay. So today we'll discuss codominance. Codominant. Remember, when we discuss codominance or incomplete dominance, always we are discussing with respect to the example. Examples are important. Okay. In which organism codominance or incomplete dominance are seen, those you need to remember. So codominance are seen in case of coat color in cattle. Okay, codominance are seen in case of coat color in cattle. You might have seen in some cows the color, there is a white along with the black patches along with it, right? So that color indicates the codominance. We will take the cross. Okay, what happened in that case? When a black color cattle is crossed with a white color, the result in F1 progeny that we get are black and white. Okay, that means both dominant as well as the recessive character shows its visible effect. Okay, so they are codominants with each other. Okay, what happened when in case of codominance a black color cattle is crossed with white again, so it should be with, we will write small p, right? So white color recessive. Okay, when the dominant black color is crossed with white color recessive, so that is our parents. So they will give its gamete. Okay, the black will give its gamete and white will give its gamete. And in F1 progeny that we get are black and white. Black and white. Okay. See, according to Mendel's law of dominance, what should have been here? It should have been black, right? Because the capital B is dominant. But in that case, that is in coat color in cattle, it is not as so. So that's why it is known as codominance. Okay. So in that cattle, the coat color with the white color, there is a black patches with it. You might have seen, right? In some cows, there is a black and white color in it. So what happened in that case? When black color dominant is crossed with white color recessive, the result that we get are black and white, which is the exception of Mendel's law of dominance. That's why what it says, Mendel's law of dominance is not universally applicable. Why it is not universally applicable? In some cases, the law of dominance do not show its visible effect, where in incomplete dominance and in codominance, right? So what you need to remember is when we cross with black and white, the result that we get in F1, what that you need to remember, the result that we get in F1 are black and white, not black. Okay, so this is the example of codominance. So what happened when in case of codominance, when black color homozygous dominant black color is crossed with the homozygous recessive white color, the result that we get in F1 progeny are black and white, which indicates that both the dominant as well as the recessive character shows its visible effect, which is example of codominance. And where we find in case of coat color in cattle. Okay, cross you have to show. Okay, that is important. So F1 we get. So if we need to find out the phenotype as well as genotype ratio, what we have to do? we have to do selfing. Selfing means crossing the F1 progeny with the same parents. Okay, that means we will cross black and white again with black and white, same parents. And what we say? We say that selfing of F1. So selfing of F1, that means we will cross black and white with again black and white, same parents. Assume that this one is male, this one is female. Okay, so to find out the ratio in F2, that means the next generation, we have to do Punnett square, right? So to do Punnett square, how many boxes we have to draw? Just count the allele number, that is 2, multiply with the same number, 2 into 2, 4. Okay, so that means we're gonna need to do 4 boxes. At the top we will write male, below we will write female. So we have B, assume this is male, okay, capital B and small b. So we will write here. There is nothing to make any crosses here because there is only 2 alleles. So capital B and small b. So below we write the female progeny. Sorry, not progeny, female parents. So B, small b, right? And you know the crosses. So what we have to do? Capital B, capital B, capital B, small b. Okay, done. Small b, capital B, I can write that way also, or I can write that way also. Okay, no problem. Both are correct. Why I'm writing that way? Because I'm putting the capital letter first. Okay, that's it, to look it better. The small b, that's it. Now find it out phenotypic ratio. Phenotypic ratio means external appearance, right? External appearance, what we see from outside. So now you find it out. The first one, that is 2 capital B are there. That means it indicates black. 2 capital B, 2 capital B. So this one is our black. The second one, 1 capital B and 1 small b. What does that mean? 1 capital B, 1 small b, black and white. So black and white. This one is also same, black and white. The last one, 2 small b are there. What does that mean? That means white. So that one is white. So what will be our ratio? 1 black, 2 black and white, and 1 white. We find. So 1 is to 2 is to 1. 1 black, 2 black and white, and 1 white. Got it? So that is our phenotype ratio. Next we need to find out this is genotypic ratio. See, in case of codominance and incomplete dominance, the phenotypic as well as the genotypic ratio that we find are same. Okay, 1 is to 2 is to 1, 1 is to 2 is to 1. It will be the same. So genotype, find out the same letter, capital B and capital B. Is there any capital B, capital B? No, there is only 1 capital B, so we write 1. Okay. Then again capital B, 1 small b. How many? 2 are there. So 2. And small b, 1. Okay. So that is our genotypic ratio. Capital B, only 1. So 1. Capital B, small b, 1 and 2. The 2 are there. So 2. Small b, there is only 1 small b, so 1. That is our genotypic ratio. Okay, that's it. Very easy. Codominance. These are the exceptions of Mendel's law of dominance. We have discussed incomplete dominance. We have discussed codominance. Okay, what happened in case of codominance? Codominance are seen in case of coat color in cattle, right? You have seen, right? In cows, in some cows, there are white color along in the skin, white color along with the black patches. Okay, black and white are there. What happened in that case? When homozygous dominant black color is crossed with the homozygous recessive white color, the F1 progeny, the first generation that we get in first generation are all black and white. But when we do selfing, that means when we cross F1 with the same parents, with the same parents, black and white, when we cross in the next generation, that means in F2 we get 1 black, 2 black and white, and 1 white. And the genotype, genetic makeup, that means internal structure, we get 1:2:1. See, very easy. Okay, so this is codominance. The question asks for 3 marks. So you need to explain first. Explain it in your one sentence. And then below you show this cross, show those boxes, and the ratios are important. Ratio you have to show. So question comes for 3 marks, either incomplete dominance, codominance. Question asks for 3 marks. And Mendel law of inheritance asks for 5 marks. When question asks for 5 marks, you have to discuss the 3 laws along with the cross, example, ratio. Alright. Okay, so incomplete dominance, let's revise it. In incomplete dominance, we take the example of 4 o'clock plant, okay, where a red color flower is crossed with white color. The result we get are all pink, and when we do selfing, the pink and pink we make a cross, we find the same genotype as well as the phenotype ratio. Same as well for codominance. What you need to remember is example. In case of codominance, we see in coat color in cattle. In case of incomplete dominance, we see the flower color in 4 o'clock plant. Alright. Okay, now again we will take next topic. Next we will take multiple allelism. That is our codominance. Next we will discuss multiple allelism. Whenever we discuss all those topics, we need to discuss with respect to example. Example are important. First we have to mention example, then we have to do crossing, and we have to show the result. So multiple allelism. First we need to know about definition. What is multiple allelism? Multiple allelism means one gene is responsible for showing many characters. Okay, one gene is responsible for showing many characters. That means multiple allelism. Now where we find multiple allelism? Multiple allelism are seen in case of ABO blood group. It is seen in case of ABO blood group. Okay. Now next important thing we need to know is multiple allelism can be detected only in a human population. It can be detected only in a human population. Okay. Can be detected only in a human population. So these are the important things we need to remember. Next we need to know is the ABO blood group was first discovered by a scientist named called Steiner. Okay, ABO blood group was first discovered by a scientist named called Steiner. You write in a proper manner sentence. Okay, ABO blood group was first discovered by Steiner, and then ABO blood group is controlled by 3 genes, and we need to know the name of that gene. It is controlled by 3 genes. What are all those genes? We need to know. We represent this gene in the form of IA, IB, and IO. Okay. So these are the genes which control ABO blood group. So these are the important features we need to know, that multiple allelism are seen in case of ABO blood group, which can be detected only in a human population, and it was first discovered by Steiner, and it is controlled by three genes known as IA, IB, and IO, whereas IA and IB are dominant, always dominant. Remember, and IO are recessive. IA and IB are always dominant. They will always show its characters. They will always show its visible characters, and IO are recessive. So next, what we need to know, we need to find out the possibilities of our blood group. Possibilities. First, we will see the possibilities of the first gene IA. Okay, we will see the possibilities of IA gene. So IA, when we make cross with IA and IB, okay, it can be IA and IA. We are not making cross, we are seeing the possibility. Okay, it can be have IA and IA, it can be IA and IB, or it can be IA and IO. Okay, so we are seeing the possibilities of first gene IA. So IA, the possibility, it can be IA, that is the gene. Okay, that is the allele. So together we call it as a gene. IA and IA, we together we call it as a gene. But one particular, this IA, that means allele. We call it the allele. So IA, two alleles together, gene. So these are the possibilities of first gene IA. So it can be IA, IB, IO. All right. Now coming to IB. Now IB possibilities, it can be IB, IO. Okay. IA and IB we have already written, so we don't have to write IB and IA. So these are the possibilities of our second gene IB. Now third possibility is IO. These are the only possibilities of IO. 1, 2, 3, 4, 5, 6. Pay attention. We are seeing the possibilities of these three genes. These genes are responsible for ABO blood group. Okay, we represent in the form of IA, IB, IO. First, we are seeing the possibilities of IA. So possibilities, it could be IA and IA. It could be IA, IB. It could be IA and IO. That is the possibilities of IA gene. And then IB gene, it could be IB, IO. That's it. Then IO possibility, it can be only IO. Now finding out the dominant one. So IA, if there is IA and IA gene, the blood group will be A. Okay, if there is IA and IA gene, the blood group will be A. If there is IA and IB, the blood group will be AB, because both are dominant. Now if there is a alleles IA and IO, the blood group will be, see, O is recessive. So dominant characters always show its visibility, so there will be only A. You will understand when you study this. Once, twice, when you do practice, you will understand yourself. Now IB and IB, so when we have alleles IB and IB, the blood group will be a B. Then when we have alleles IB and IO, the blood group will be again B. Okay, because O is recessive. Now when IO, the blood group will be O. Okay, these are the possibilities that we can find in case of ABO blood group, which is controlled by three genes. The possibilities can be A blood group when we have two alleles IA and IA. It can be AB blood group when we have two alleles IA and IB. It can be A blood group when we have alleles IA and IO. When we have, it can be B blood group when we have IB again same for those. Okay, now we need to find out the phenotype and genotype ratio. So phenotype, there are 6 phenotypes we find. 1, 2, 3, 4, 5 and 6. No. There are 4 phenotypes we find. See, capital A and capital A are same. So 1. Capital AB. Okay, capital AB 2. Capital B and capital B 3 and O 4. So phenotype, we have 4 phenotype. Genotype, there is total 1, 2, 3, 4, 5 and 6. So 6 genotype we find. Okay, so they are known as multiple allelism. 1 second. Phenotype. Now, phenotype, we have 6 phenotype, and genotype, we have 4 genotype. Okay, total phenotype, the external appearance, that is the external appearance phenotype. So we have 2 capital A, 2 capital B, A, B and O. They are the external appearance. So we have total 6 phenotype. Genotype, we have 2 capital A, so 1, 2 capital B, 1. So 2 capital A 1, 3 and O 1, 4. So we have 4 genotype. Okay. Practice, please practice. You will understand. So that is multiple allelism. So where we find multiple allelism? Multiple allelism we find in case of ABO blood group, which was given by a scientist known as Steiner, and it can be detected only in a human population. Okay, not anywhere else. So it is controlled by 3 genes: IA, IB and IO. When we see the possibilities of first gene IA, so IA can be IA, B, IO. So there will be A blood group, AB and A blood group. And when we see the possibilities of IB, we find 2, IB and IB and IO. So that will be a B and B blood group. And the O possibilities, we find only that one. So when we see the phenotypic ratio, we find 6, 1, 2, 3, 4, 5 and 6. When we find genotypic ratio, so capital A and capital A 1, capital B and capital B 1. So 1 plus 1 2, AB there is only 1, 3, O there is only 1, so 4. Alright, so this is multiple allelism. In next topic we will see pleiotropic inheritance in next video lecture.